and it will be given by Dr. Mark Lyte, and I'd like to just give you a short introduction. Dr. Mark Lyte is a full professor with board certification in clinical laboratory medicine in the Department of Veterinary Medicine and Preventive Medicine at Iowa State University. And today, he's going to talk about some of the study he has done on microbial endocrinology, and his title, the talk title, will be Stress and the Microbiota Gut-Brain Axis, Implications for Poultry Gut Health, Infection and Inflammation in the New Era of Antibiotic-Free Production.
Thank you very much, and I would very much like to thank the organizers of this meeting for inviting me. As you probably saw by the slide on my background, I'm a clinical person. I'm a board-certified laboratory scientist. I've run hospital labs up to 900 beds, laboratory, level 1 trauma. I've trained surgical residents in research. And so I'm coming at this with a different set of eyes. And what I want to show you is that ever since coming to a vet school, because basically I have been at clinical for over 35 years, and I reached a point where I'm trying to study the microbiota. I'm trying to study what determines infection, maybe behavior, and using humans, where you only have access to their poop, and not many people will agree to sacrifice part of their intestine for me to study, and you have mice, you have no in between, and it became very difficult. So about 3 years ago, a little less, I got an opportunity to go to a vet school and be involved with animals. And what I've been learning is this, that what affects cattle and infection in their lungs is the same evolutionary pathways of the microbiota that affects pigs, affects patients in the hospital, which is what I worked with for a long time, and affects poultry. I've only been doing poultry for a few months. I'm very new to this. But what I want to show you is that by looking across common evolutionary ways the microbiota interacts with animals, you'll have ways of then treating your animals and eventually, for me, also getting to humans. So the purpose of the talk is not going to review a literature, but I'm going to introduce to you a new perspective that may lead to innovative ways of utilizing nutrition to help poultry health. And I'm going to give you a longer perspective. I've been in the field for quite a few years and the rest, and it's a personal perspective of mine. I'll get into it a little later. Some might call it skeptical, but you're hearing it from others. But first, I want to start with this way. For those of you who took microbiology, you learned about bacteria in your shaker flask growing 1 to 2, 2 to 4, 4 to 8. And that's how you learned about them. I call it dumb bugs. They just grew. They didn't have any type of social structure to them or anything else. I'm going to introduce to you today bacteria in a very different light. That as neuroendocrine organisms, that they have a neurochemistry to them, which turns out to be exactly like ours. And because of that, there's an evolutionary vantage point that I'm going to be utilizing throughout my talk. And because of this evolutionary neurochemistry-based background, that what I'm going to show you is that then there's a complex interaction between the host, diet, and microbiota that's going to influence disease progression and health in poultry. And what do we really know about poultry feed? So I'm going to take you back beyond even poultry to the ingredients that go into your feed and show you that you don't know all that's in it. The concept of this evolutionary neurochemistry-based understanding of bacteria and how it interacts with animals has been termed microbial endocrinology. And we're at a critical juncture of using the microbiome. You've heard a lot of talks about the microbiome in health, right? But I'm going to argue, and I'll do that at the end of the talk, I'll make sure I leave time to do that, that the microbiome is in a bubble-related phase. It's in a phase of where it's expanding like the human genome did, and there are going to be holes poked in it. I'm seeing a lot of the same issues come up in poultry now that I saw in the human development of the microbiome and things how we're moving on to microbiome version 2. The poultry microbiome looks like it's in version 1 of what we already found out were problems with how we looked at the microbiome in humans and animals. Animal nutrition offers a unique opportunity to explore microbiome health interactions, especially now given the removal of antibiotics. And one thing I haven't heard from any of the speakers, I must say, is that they're presenting the microbiome as lists of organisms. We're forgetting something, that the microbiota functions as a community. I think of it often as a community of people, such that they live together, they function together, they form their own specific ecological niches. And if we're just going to look at diversity and listing bacteria down 1, 2, 3, 4 like this in a row, we're not going to understand how they interact in the animal. Because besides the fact that a lot of the microbiome data that you see is from feces, but it doesn't tell you what's in the animal, in the gut, where it has to have action and then help out the physiology. And that these communities don't function by themselves. They are bidirectional and interact with the host. With the removal of antibiotics, everything's changed. So there's tremendous new opportunities to use the microbiome, but it's going to be how you, in your research, your company uses it to approach it and forge new paradigms. You've seen things like this before. This is on the human side. Of course, gut microbiota seems to affect everything. Everything that people have looked at, it affects acne, everything, hair growth. It is a long list. A lot of this type of hype also belongs and applies to poultry health as well. And in a way, research is all well and good, but at the end of the day, what does looking at the microbiota and how you look at the microbiota mean for the poultry industry? So what are the take-home lessons for your particular company that you're interested in the interaction of the microbiota with a specific product or practice? So, nutrition. There's a really large literature on nutrition in the microbiota. It goes back decades. I'm not going to review that. But what I am going to do is look at how the microbiota interacts with poultry from an evolutionary-based approach to link components of the gut, immunology, microbiology, and what you see happen to your animal. So neurochemistry is going to be a common evolutionary language to link everything together. But I will say right away, microbial endocrinology is one mechanism of a whole host of mechanisms that are being proposed. I'm not going to say it's the only mechanism. It is one of the mechanisms. And it's also the realization that when anything is ingested in your animal, it's really not in your animal, as was pointed out. It's outside. It's in the gut. It has to then get into the animal. But the first thing that comes into contact is the microbiota. And so we're going to go over the food and what's in food, actually. So as I said, neurochemistry is a common evolutionary language. So what's in poultry feed? This is what you typically show to be in poultry feed, all of these ingredients. You'll show selective nutrient, protein, carbohydrate, fat, and the rest, but what I'm going to show you is there are other things in there. There's a neurochemistry that you haven't known about. So what else could be present that influences the animal either directly or indirectly? These feeds, when we've been looking at them, differ in neurochemistry, although they look to be the same by the other parameters that I just showed you. So microbial endocrinology defined. It is the union of microbiology and neurobiology brought together, two fields you don't think often come together to form microbial endocrinology. And that has implications for disease and behavior, stress and nutrition interact, and that's going to have poultry health. And evolution is going to be a theme throughout. So you see things like this, raising happy chickens. Where can you really see neurobiology and microbiology come together? Well, let's go in a way to cows. This is actual product that is sold in Ireland. It says, lullaby milk, simply sleep, a sip, and slumber. Because our cows are milked before dawn, lullaby milk contains higher levels of melatonin. And because they produce this melatonin, you're going to feed it to your kid, and what's going to happen? It's going to regulate your sleep cycle. But here's a question. Melatonin in milk, it's going to get into the gut, and you're saying it's going to affect the brain. But it's not the brain that is first seen by what is ingested. It's the bacteria. Here's something interesting. This was just published a year and a half ago. This is a plate, petri dish, which they put one bacteria from the gut of a human in there. And it grew such as this. They then added 1 nanomole, an incredibly tiny amount, 1 nanomole of pure melatonin. And the bacteria responded and pulsed in a circadian rhythm. We don't know why it does that or how, but it's a circadian frequency like our sleep cycle in the gut. But you can ask, why is a hormone produced in the milk that helps out our sleep cycle? You think the cow was designed that way? Certainly not. It must be another role why it is, because melatonin is not secreted in milk to benefit human population. Is there an effect on the calf microbiome? It's an example of how the fields of microbiology and neurobiology actually come together, and you can see these things. Which brings us to how all of this started. Stress affects immunity, affects disease. You were all taught that stress is bad for you and affects immunity and disease. I'm going to show you, is that really the complete story? It's bad for you, and that leaves you open to disease. And I was really happy when I came into the poultry health because I've been doing mice as well as humans for many years. And a lot of my work has been in mouse work. And I've been doing something called social conflict. And then I found out that, wow, chickens undergo social conflict as well, especially when they're free range. So what was I doing? I was taking a mouse as an intruder, putting it in this residence cage, trying to understand. This was back in the 1980s, actually. And what would happen is you put an intruder mouse into a resident cage, this big guy right here, they fight, and this mouse gives up. It goes through what's known as defeat. And we pulled this mouse out and said, wow, this mouse must have a suppressed immune system. Why? Because it's stressed. And what do we find? Exactly the opposite. We found no change in all the parameters of immunology you would think of, but that was lymphocyte-based, because that's adaptive. No one was thinking that the innate system, which is concerned with bacteria killing, would be changed. That's why we call it the innate. So we were able to show in the 1980s that phagocytosis of the stressed animal was increased over 500%. And many people have replicated this as well in mice and other systems. So a point, stress, acute stress, not chronic. Because acute stress is why we have fight or flight hormones, right? Adrenaline, noradrenaline, otherwise known as epinephrine and norepinephrine. Chronic stress is something completely different. Evolutionarily, chronic stress doesn't really exist. In the way back 10,000 years, you were always faced with a fight or flight situation. You either made it out or you didn't. It didn't make any sense if you were presented with an infectious challenge that evolution would say, well, you've been stressed, here, go over and die with your immune system. It made more sense that stress acutely said to your immune system, challenge coming, you better get up. And that's what we saw. Chronic stress, if you were chronically stressed back then, you usually became nutrition for other animals. So, with that in mind, what did we do? We took these animals with such great immune systems to kill bacteria, and we fed them bacteria perorally, and we said, wow, you're going to kill them all. And what we found was exactly the opposite. Animals that were defeated with a heightened immune system died faster with infection. How is that possible? That's when I gave up immunology and became microbiology. How does it make any evolutionary sense? It makes no sense. Increase the immune system, I give you bacteria, you die faster, right? So what I want to ask you is, who's really stressed here? And let me posit to you to have you think differently. It's the poor bacteria. Think of your chickens that develop necrotic enteritis because of C. perfringens and they get infected. They're not getting infected with hundreds of millions of bacteria the way people dose these animals. They're picking it up in the litter somehow, and they're picking up a few spores or a few vegetative bugs that get in there, and they find themselves in the intestinal tract facing communities of bacteria that don't want them there. They are stressed. Do you know how many E. coli 015787 bacteria from a contaminated hamburger patty takes to kill or to cause a 4-year-old to lose their kidney because they've undergone hemolytic uremic syndrome? You know what the effective dose is? 5 bacteria, 5 CFUs. How are 5 bacteria able to do that? I'll tell you one thing, they're not happy bugs. They came out of a cow, they were ground up in the meat, they were frozen, you tried to cook the burger, someone ate it, it went through the gut, hit stomach acid, but yet those 5, and maybe they're down to 3 or 2 now, survived. Do you think your immune system sees 2 bugs? Nope, it won't see it. They have to survive now in a hostile environment. They are stressed. So who does this make sense? It makes sense for the bacteria. So stress, immunity, disease, took immunity out of the equation and said, how do microorganisms know where the heck they are? Are they neurochemical responsive? So before I show you that, I'm going to take a step farther back on the evolutionary cycle to tell you plants, the basis of all of your feeds contain neurochemicals. You don't think of it that way, but they do. Potatoes, for example, when they're stressed, they make catecholamines, norepinephrine, and dopamine. They make, some of the same neurochemicals in fight or flight. What are potatoes doing with our hormones? Bananas. If you were a group of cardiologists back in the 1950s, I would be telling you, you better stop feeding your patients bananas. Why? Because as the banana browns, it gets more and more neurochemicals. I mean, large amounts of neurochemicals that can affect the heart. It wasn't until 1958 when you could publish in Science by peeling a banana. And that's what they did. They got into science by peeling a banana, and they saw that all of the neurochemicals were in the peel and not the pulp. It became okay to eat bananas again. But nobody asks a simple question. What the heck is a banana doing with neurochemicals? And why is it browns? Does it actually get more of them? These are pure neurochemicals. Tribal pulses. You've all seen the things on TV documentaries where in parts of Africa and other countries, they beat these seeds on hot, flat rocks, make a bread, and then the bread is eaten, right? Well, after World War II, some of the first people into those regions were, of course, the British. They came back to England with heart conditions. They couldn't understand what was happening to these people until they looked at what they ate, the nutrition of these tribal pulse seeds in Africa. They found 8 grams, not milligrams, pure 8 grams of pure L-DOPA. L-DOPA is the neurochemical that is needed for treating Parkinson patients to have them make dopamine, to have their brains work properly. What is a tribal pulse doing with L-DOPA? Tomatoes have neurochemicals as well. Even when they're stressed, they make stress neurochemicals, our neurochemicals. Again, we think of it from our point. Cheese has tyramine. I often bring this up. Pigs, truffles, and microbiology. What's the connection? Why does a pig hunt a truffle? The answer is easy. It wants to have sex. Why? Because that's a female. When the pig finds the truffle, it stands up to be mounted, because in the foaming saliva of the wild boar, of the male, is androsterol. And so you can start to ask a question, what the heck is a truffle doing with the pig's sex pheromone? That's not the proper question. The truffle came underground before the pig. How did that get into a pig and be used as a sex pheromone? We don't know. So now let's get back to bacteria. When you look at bacteria, it's been known for decades that they contain neurochemicals. If you ate yogurt and had lactobacilli in it, it produces gamma-aminobutyric acid, the primary inhibitory neurotransmitter in your brain and in your poultry's brains. Catecholamines, the complete biosynthetic pathway for stress, fight or flight hormones, are made by E. coli. We don't know why. They make it exactly the way we make it. Protozoa make it, fungi make it, as I said, but that's all well and good. So plants have these neurochemicals along this evolutionary pathway, bacteria have them, okay? But does it matter? Do they interact? And what I'm going to show you is that these concepts work both for chickens, Campylobacter jejuni, and patients in the hospital bed, Staphylococcus epidermidis, and from the microbiota gut-brain axis, because you want to raise happy chickens. This is actually the first time it was done, and I was so happy and I was so excited back in 91 actually, that I actually used a Polaroid camera, and I have to explain that to students what a Polaroid camera is. What I did was take bacteria and I put it into a difficult media for them to grow, Yersinia enterocolitica, which is a gut pathogen, as you probably know. I didn't put it in luria broth or any of the other ones that are typical microbiological media that we use to study growth, because that's not the way bugs survive in real life. They aren't fat, happy bugs that grow in the tissue culture flask and that you then get all your omics technologies out, because the way they have to survive, that's different. They have to survive in a more hostile environment, and that's what we put them in. And when you see you put it in here, you can see there's very little growth. If you put epinephrine in micromolar amounts, right? Micromolar, 10 to the minus 6 of a gram. You put that in, nothing happens. There is no epinephrine in the gut. There are no neurons that synthesize epinephrine. They synthesize up to norepinephrine. Look what happens if you do that. Within 24 hours, you have over 10 to the 8th logs of growth. Hundreds of millions more bacteria being made, even though there's no nutrition here to support it. This isn't supposed to happen, but it did. If you then take bacteria and you try to put it in a mouse, these Yersinia, they don't infect. Everyone knows that who uses Yersinia. You can't get them to affect. Simply expose them to norepinephrine, and look what happens. They now grow faster, and they begin to invade the intestine. It reduces the infective dose more than 50,000-fold. You need that less bacteria. There have been studies before using Clostridium back to the 1940s, we found, that actually for not doing it the way we did, but show that you could reduce the infective dose by 1 millionfold needed to cause infection. So, poultry and Campylobacter, bring it back to that. Just as you think you're the first, 1976, we found a paper where people looked at Campylobacter. Now, clinically, Campylobacter is a disease which is a huge issue of infectious disease. If a patient has Campylobacter-associated diarrhea, in the clinical lab we have to know that coming in, because otherwise we don't put the bacteria in the sample in what's known as microaerophilic conditions. We have to treat them really gingerly. We can't just leave them on the bench. But you know in poultry, there are no microaerophilic conditions. When that Campylobacter comes out of the bird in feces and then infects another bird, it's out in the air. How does it survive? It makes no sense, again. So what did these people do? And they never listed a reason, but they took the Campylobacter, put it on a plate, and it wouldn't grow in the air. Everybody knows that, still the same. They decided to add a little norepinephrine to the plate for some reason. Look what happened. Started to grow. They added more norepinephrine, really started to grow. They made Campylobacter go from microaerophilic into aerobic. Okay, so now back to humans. This is 2003. And so one of the biggest troubles in the clinical hospital and surgical intensive care are patients get infected in intravenous lines with staph epi, the skin bacteria you have on your hands right now everywhere over you. How is that possible if the nurse or whoever puts in the catheters cleans everything off? How can that happen? It's been unknown for decades. Over 50 or 60 years until this study, which was the cover story of Lancet, which finally explained it. This is what the inside of a catheter looks like on scanning EM after you put six staph epis on it. You don't see anything, that's background noise. Simply put the same drug that goes down the catheter into the patient, which is dobutamine, which is an analog of dopamine, a catecholamine, to keep heart function and kidney function going. What happens to those 6 bacteria in less than 30 hours? That's what happens to them. They grow into this massive biofilm with channels for antimicrobial resistance, all the things that contribute to it. So it was finally the explanation why patients were getting infected. It was physician do no harm, it was giving the drug that was activating the bacteria. And there have been calls ever since then to find alternatives to it. They haven't been able to find alternatives to dobutamine. So there's a common evolutionary theoretical thread here between microorganisms as the host. Cell-to-cell signaling that poultry use, that we use, first may have evolved due to late horizontal gene transfer from bacteria. So bacteria have had it before us. Is it something that they use for form community structure? We don't know, but we were looking at it in the lab. But it's important to understand that microorganisms, such as those in the gut, don't simply rely on the traditional nutritive energy sources that are in your feed and that you constantly check for. They look for signals to know where they are and tell them what to do. It means that there are direct neuroendocrine bacterial interactions, and bacteria are not dumb bugs. Okay? I'm going to skip over the inflammation for a second to get to the gut, where neurochemicals and bacteria meet. We heard a very good talk just before on intestinal development. One thing I would like just to add to that very good talk is that the realization there's a neurochemistry to the gut. In fact, physicians and scientists had always thought your brain, your lungs, and all the rest were where neurochemistry was, where you made all these neurochemicals, or liver, wherever. It wasn't in the gut, in the alimentary canal. Well, guess what? They've looked at pigs, humans, other animals, that the production and metabolism, norepinephrine and dopamine, is over 50% of the total body occurs in the gut for reasons we don't know. Your stomach is a dopaminergic organ. It's making dopamine as we talk, and as I talk and as you listen. We don't know why, it just does. Within the gut, you are putting out neurochemicals into your lumen. You're making serotonin right now as you sit there. You're going to poop it out. No one knows why you're doing it. We don't understand it. No one's looked in poultry, by the way. And then there's food, the dietary sources. So the foods are coming in, and they have neurochemicals as well. We're going to talk about a very interactive environment now, known as the microbiota gut-brain axis, where if you've been seeing what I said, food, the nutrition, provides substrates, neurochemicals themselves, or the substrates to make neurochemicals. When you ingest it, when your poultry ingests it, it first comes in contact with the microbiota. They then do something with it. They either then can make neurochemicals themselves, which are uptaked into the portal circulation, or they can hit the enteric nervous system. What you haven't been hearing a lot about is the innervation of the intestines. So I'm going to go into that because it's important for you to know that in poultry, there's not much known at all. So there's an enteric nervous system that you all have. That links up to the brain. Believe it or not, the first nerves ever found were not found in the brain or wherever. They were published in the 1800s in the intestine from dogs. So this enteric nervous system, which innervates your gut, 500 million neurons, that's as much as your spinal column, links up to the brain. The main trunk is the vagus. And it's a direct interaction where you hear about gut to brain, okay? But we're going to bring in bacteria into this. So they're now communicating with the brain, and that affects behavior and cognition, and maybe we've proposed, and others are also doing it, that they're also telling the brain what foods they want. Because as you take off foods, they die if it has a substrate they want. Why would they want to die? Is there a way that they're actually communicating back to the brain? And here's an important point, and this touches on other lectures you've heard about the microbiome, and it also applies to mine. So again, I'll be very honest with you. Correlation and causation, one of the banes of microbiome research. Just because something in the microbiome is changing and something else physiologically happens, that's not causation, that's correlation. Two things happen at the same time. Microbiome version 1. There are tens of thousands of publications of that. How much of it actually is made into clinical practice? As a gastroenterologist once told me, goose egg, 0. Okay? Here is innervation. Your gut, as well as the chicken, from the little we know that's been done in the chicken, is differentially innervated. That feeds back to the brain. That means there's the microbial biogeography, and you can't get that understanding from looking at fecal specimens. You have to go into the animal and look at it. So what do we know about the innervation of the alimentary canal of poultry? Exceedingly little. And here's the reason why. This is a villus. There are nerves that go to the tip of the villus. We don't know why they're there. We don't know what they're doing, but they sense the luminal contents, and information flows, as I'll be showing you. To show you how powerful this was just done 2.5 years ago. They took a trigeminal neuron from the brain of a mouse, and they then took a gut cell, an enteroendocrine cell, and put it in a petri dish next to each other. And after a while, what did these two cells separated from each other do? They made a nerve to connect up and talk to each other. Amazing stuff. What are the consequences of all this thing going? This is a picture of what's known as an elevated plus maze in mice. It's how all your psychoactive drugs, antidepressants, start their life to treat people. Basically, any of you who have had a mouse in a garage or anywhere else, you know it runs and stays in the dark areas and is really tough to catch, right? And so this is a device that's all used in the pharmacological industry, which is you take a mouse, you put it right here, and you let it ask a question, make a decision. Does it go into the closed, dark, protected arms, or does it go out and be a brave mouse and venture out on the open arms? If you can make that mouse go on the open arms, you run out and buy your first BMW and Mercedes, because you found something that is an antidepressant. It basically changes anxiety levels. This is what it looks like when a mouse is running. There on the closed arms, it stays there. But if you cut that connection from the gut to the brain, guess what happens? The mouse is now brave. Changes its anxiety. We don't know why that happens, but it does. There's direct communication going on. The take-home message from the authors that they publish emphasized an important role of afferent visceral signals in the regulation of emotional behavior, also known as gut feelings. So they finally, in 2014, proved what your mother always told you, that she has a gut feeling about something. That's what they proved. So where have we come from? We talk about probiotics and microbiota gut-brain axis. This is not new. This is not new. Here's probiotics in 1914. The control of man's diet is readily accomplished. Mastery of his intestinal bacteria flora is not. There's cases of malaise, lack of ambition, every effort in life is a burden, depression. How do you treat it? Bacillus bulgaricus, what's in your yogurt today, one of the main probiotics used. This whole concept of microbiota has been known for a long time. The gut of the poultry has innervation. We really don't understand all of it. But the first modern era demonstration of the microbiota gut to brain was in my laboratory back in 1998, where we simply put a novel bug into the alimentary canal of a mouse. Not one that would cause any problems from pathogenicity. It didn't invade. It simply was dividing and going through the animal. And what did we find out? We found out that the mouse became anxious. They developed anxiety-like behavior and stayed in those closed arms more. And when we actually looked in their brains, what happened? When you put it in the gut, and it didn't do anything but simply pass through and divide, nerves in the brain associated with behavior, nucleus tract solitarius, paraventricular nucleus, all these dots begin to light up. These are activated neurons by what's known as CFOS staining. So the brain was monitoring what was in the gut, microbiota gut-brain interaction. So all of this, I'm going to put it together, involves a long evolutionary pathway where the neurochemicals in your birds, in man, first evolved in bacteria. That means there's a symbiosis here, doing both where the bugs are and the brain that can interpret those signals. It means nutrition plays a powerful avenue here with which to treat them and modulate that behavior. And just to quickly show you that if you simply change nutrition in a mouse, you can change their behavior. So we fed mice meat. We compensated and equalized for energy and all the typical things. But what we wanted to do was change the microbial diversity. And this is old 454 pyrosequencing data to show you we changed the diversity between a beef diet and a control diet. And we basically asked mice to find a hole, an escape hole, on what's known as a Barnes maze. They have a lot of holes to go to, but you have to train them to remember where to go. And an animal which is not stressed remembers to go to that hole really fast. If you stress a mouse, that's where they start running around trying to remember where the hole is. However, so here you see how much an animal takes running on that area to find out how long it actually runs. If you stress it, it runs more. If you feed it beef, it doesn't change between here and the standard diet, which is the vegetarian diet. But as you increase microbial diversity, what happens? The animal, it abrogates the effects of the stress, and the animal remembers better. So returning back to the species at hand and central question, what's the way forward? What's science telling us? Where do we need to go? What I want to posit to you today is that the study of microbial endocrinology will be crucial to improving poultry health, considering what's in your nutrition beyond what you've always been taught to consider and probably are doing at your companies. Mechanism. Without it, we're groping. Again, correlation and causation, the trouble that's been deviling the microbiome in human health. Any mechanism, I believe, at some level has to include neurochemistry. And remembering it's a very long way from the gut to the brain. And using microbial endocrinology as an evolutionary-based mechanism. So in the last 5 minutes I have, I want to tell you why I think the microbiome is in a bubble hype. Okay? Here's from the New Scientist published about a year and a half ago. Microbiomania, the truth behind the hype of our bodily bugs. At that point, we were having the White House's latest moonshot was studying the microbiome. And what they said was, there's a gap between the promise of the microbiome and the current science. It has been filled with misconceptions, snake oils, and hype. Microbiomania. Microbiome research is incomplete. We simply do not know what many microbial genes do. Let me ask you a question. All the bacteria you've been seeing in your sequence, or if you see it and you do this, do you know what all the genes do, or how much percentage of the genes what they do? Escherichia coli, which is the most understood organism in the world, what percentage of the genes do we know what their function truly is? 60%. 40% we don't know. I myself, when I go through a lot of these microbiota data, I keep coming up unknown function, unknown. And what do? We go to what's known. But in some of the ones that you're looking at and you then get on your sequencing, you might know 3 or 4% of what it is. 90+% you don't know. How are you going to treat that data? You then go to the next step and you go to metabolomics. And you do an untargeted metabolomics, which I've done many. And what you find out, because I'm clinical again, I need it to repeat. When a patient comes in and they think they're in a diabetic coma and they send it to the lab and I'm the guy doing it, I better be sure if I tell them it's 10 mg per DL or 400, it's a completely different therapy then. You can kill the patient the wrong way. But metabolomics, it's semi-quantitative at best, and it doesn't account for matrix, the matrix of the fecal specimen, because you get ion suppression and ion enhancement, and things will shift. And there aren't standards for all 4,000 or 5,000 of those things that you get back in your report. You put that, and we only understand 4% of metabolomics. So we take what we know, we put it what we know in the others, and we come out with this stuff. Does that make sense? And also I want to change a misconception. You heard about fecal microbial transplant, and it started in 2009. That actually is incorrect. The first study of FMT and use in pseudomembranous enterocolitis, which is caused by Clostridium difficile, was 1958, where Einesman in Colorado gave a fecal slurry to a patient and cured them. So FMT did not depend at all on the microbiome. Neither did the other microbiology-based therapy that we use to treat ulcers, Helicobacter pylori. That was done before the advent of the microbiome. So what are the lessons for your particular company? What are you paying for? So I should take a second, I have a couple minutes left, to tell you why is this happening now? Why now? When I was a student back in the 70s and 80s, microbiology was dead, was dying. We had all the antibiotics. We don't need it anymore. But then we had environmental microbiologists looking what was in the sea, developing techniques, and eventually came up with these machines, these sequencers, that gave us data, loads of data. I always see people get these loads of data, 10, 100 million sequences. We get these OTUs. Usually we throw out 30, 40%, they're not good. I keep asking the bioinformaticians, why are you throwing it all out? Oh, our program throws it out. Explain it to me. They can't. And you get this. But what does this all mean? Again, I'm being skeptical here. I'm being a bit of a contrarian. I use this all the time myself. If you look me up, I publish on this, but I want to look at it with a clear eye and be careful of the hype. The technology is changing. So much of what we did was 16S before you saw 454. Now it's Illumina, 16S. It's going to what was shown before by Professor Duray, whole genome shotgun sequencing. But there's going to be something else. Finally, I'll finish on probiotics. Why this one? Because I was asked, some people have come over to me and asked, in their company they use probiotics. One time it works, one time it doesn't. Why? Because you forget something critical. Why this one, why not that one? When you buy an ingredient from a supplier and it's copper, whatever it is, selenium, I don't care what it is, you can test it yourself and have an assay and know it's there. When you buy a probiotic, what are you buying? You're buying that genus and that species, but you're not buying the physiological state of the bug. You're simply buying the bug. And how are they grown? They're grown in up to 10,000 liter vats, 2 stories high. Do we know physiologically a measure to say that bug grown in that 10,000 liter vat, which has undergone how many millions of generations, is physiologically going to do the same thing you want it to do when it gets into your poultry? We don't know. How about yourself? In that vat, growing those bugs, right? They undergo more evolutions than man has undergone in your evolution. Are you the same as at the start of evolution? No. Are the bugs the same? Probably not. So the companies have to evolve to give you physiological measures, quality control, to know what's going to work in your system, not just genus and species. I have actually proposed that they function as neurochemical delivery vehicles, and I'll finish with this. Let me just finish with this. This one thing, whenever we think they're first, we're usually not. I always teach my students this one, The Psychic Life of Microorganisms, a study in experimental psychology. In other words, this person called Alfred Binet said that bugs actually have a psychic life and they affect us, published in 1903 in English. And I often ask crowds, who is Alfred Binet? Because Alfred has touched every one of you sitting here, and the French should have the answer to this, because it's published from the French in 1888. Alfred Binet, the father of modern IQ testing, said, bugs have a psychic life. Thank you for your time. Thank you.